Hello, thank you for joining me in this presentation. In this presentation, I will be talking about how we use exome sequencing to identify rare coding variant in novel human mouse orthologous genes in African individuals diagnosed with non-syndromic hearing impairment. Hearing impairment can affect the external hair, the middle hair, or the internal hair. As the case may be, genetic factors contribute to hearing impairment. Studies have shown that 75% of non-syndromic hearing impairment are associated with mutations. These mutations have been identified mostly in GJB2 gene and other 150 genes. No mutation in these genes in non-syndromic hearing impairment in African populations have not been identified or understood. Investigating mouse hearing genes can be instrumental because mouse studies were used for establishing hearing mutation in other populations. The purpose of this study was to perform exome sequencing on Illumina platform to investigate pathogenic mutations in 34 novel mouse hearing impairment genes that were recently identified. Patients were recruited from Cameroon and South Africa. There were familiar cases among the patients and also sporadic cases. The cases were mostly prelingual, that is early onset, and there were no other clinical signs associated with the patient. After the sequencing, we performed some analysis to check the quality of the sequence data and also for the annotation of the sequence data. The result shows that the Cameroon and the South African sequence data were of good quality. And using other algorithms to annotate variant, we identified specific variant in different regions of the genes. However, we focus on the 25 missense variants that were identified in the study. Further analysis, we classify 4 variants in 4 genes: OCM2, LRG1, MCPH1, and SPNS2 gene. The MCPH1 gene was identified in homozygous state. Further analysis on the MCPH1 gene shows that this variant is located in a position that could affect the structure and the post-translational function of this gene, and including other genes that are related with the MCPH1. This was the first investigation of human mouse orthologous genes in African population that found homozygous variant that could explain hearing impairment in patients, and 3 other variants in heterozygous state in other patients. Thank you for listening.